Good morning. It's wonderful to be here today. It's good to see so many people in attendance. If I had it my way, I'd much rather tell you about stick bug evolution. I think that's a much more interesting topic anyways. Nonetheless, apparently, there has been a lot of discussion on this issue, and that's where I come into play. And hopefully I might say something today which might bring you some greater insight. Now, I've had a cold for the last 2 or 3 days. I hope my voice will hold out, but we'll just kind of see how things go. Well, recently, as most of you are aware, there has been a proclamation put out by critics suggesting that DNA evidence demonstrates that the Book of Mormon is in fact not true. They consider that Mormonism has been dealt a death blow. They suggest that concrete, scientific DNA evidence, okay, it's not just scientific, it's scientific DNA evidence, proves that the Book of Mormon is false, and furthermore claim that there has been a hush over the BYU campus. And the reason why they say there is a hush is because the feeling is that there's been nobody here who is willing to respond. They think they have an argument which is so strong, so concrete, so well founded in science that no one would ever dare challenge them. Well, I think what I'm going to demonstrate today is that, in fact, there are a number of us who have expertise in this area, and the reason why there's been a hush is we simply didn't think the arguments were good enough to waste time responding to them. And I will discuss this in a bit more detail. We are the modern Galileo. Hear us roar. Okay? This is in reference to the notion that, much like Galileo, they have come up with a scientific discovery for which they are being persecuted by the church. And so it makes for a wonderful story. And this is what has been in the news. The notion of we've got objective science over here, we've got dogmatic religion over here, the religion is telling us to keep quiet. Aren't we sad? Okay. Well, let's look at the critic's argument. Okay. And let's go through and we'll just dissect this in greater detail. But basically the argument goes something like this. The Book of Mormon predicts that the descendants of the Lamanites will carry DNA characteristics of the Middle East. Further, that Native Americans are the direct genetic descendants of the Lamanites. They point to research which suggests that Native Americans do not exhibit Middle Eastern genetic markers. And finally conclude that the Book of Mormon is false. Now, when you're taking this line of argumentation, where you're using DNA evidence to infer something about the past, then you have moved into a realm of science which is encompassed basically by 3 areas. The first is phylogenetic systematics. And this is the branch of science which uses DNA and other information to reconstruct phylogenies or genealogies among different species. And this is primarily where my expertise lies. There's a second field called population genetics, and population genetics is interested in what happens to DNA within populations over time. And we're fortunate to have Dr. Crandall, who will be here later, whose expertise is in population genetics. Molecular evolution. Molecular evolution is the study of how molecules, particularly DNA, change over time. And we have an expert on the panel to speak about that, and that's Dr. David McClellan. But what I want to emphasize is that all of this is a subset of evolutionary theory. And what we are talking about today essentially hits this area where these 3 disciplines overlap. Now, my colleagues and I are cross-trained in all these disciplines. We have different emphases in our own particular research. But I find it, if I can take an aside, I find it extraordinarily ironic, delightfully so, that evolutionary biology is coming to the defense of the Book of Mormon. That is not something you would have predicted a few years ago. The other point to make is that despite what you might have been hearing recently on emails floating back and forth, Brigham Young University has one of the strongest systematics programs in the United States. We are recruiting graduate students and postdocs from all over the world. I have some postdocs here today which can attest to that. And we have gained a reputation within scientific circles as being an outstanding place to do this sort of research. And so this notion that it's sort of a backwater place to look at DNA issues is not really correct. Well, let's jump ahead. Let's talk about the critics. Now, what I want to point out is that the critics have not been formally trained in molecular evolution, molecular systematics, population genetic theory, or evolutionary theory, or at the very least, they are not actively doing research in this area. They are not formulating specific testable hypotheses. They're not collecting or analyzing data. They're not publishing in standard molecular journals where people who are best equipped to evaluate their conclusions can actually respond to them. They ignore the implications of modern historical research on the Book of Mormon. And what I want to point out is that they are not actually performing research. They talk about all the DNA research they have done on DNA. They haven't done the DNA research. The DNA research has been done by legitimate population geneticists who were looking at specific questions where their research was designed to get at these questions. And what the critics have done has been to look through the scientific literature and pick out things which support their point of view while conveniently ignoring some complicating factors. And we're going to talk about that. Well, let's talk about the nature of science. Science is hypothesis-driven. Okay? Those of you who think it's not have not been in science very long. Data collected without specific hypotheses and experimental design are of limited scientific utility. Now, I've got my brother up here who's sort of in the front. He is a professor of statistics at Brigham Young University. And he tells me that on a continual basis, week by week, he has scientists who walk into his office and say, I've collected all this data, what does it mean? And he says, well, nothing, because you did not design an experiment. Is that right, Dave? Give me an amen. Okay. Okay. Wanted an amen, but that's okay. And this is an important point, and we recognize this as we approach scientific problems, that science is in the business of making hypotheses and testing these. So you need to know what the question is, you need to know how you're going to test it, and you need to know the limitations of the results. And finally, and this is a critical issue, and this is why science operates, and this is why science grows and is as productive as it has been, it's this notion of peer review. If you are not publishing where other scientists can review your work, and if you are not seeking out grants from funding agencies where you can have your peers look at your scientific design and evaluate it before you even get into the project, then you are not doing what is regarded as the highest quality science. Now, some people think that you've got science and non-science, but in fact, those of us who are scientists recognize that there is good science and there is science which is not so good. Well, this brings up this issue then. Is the authenticity of the Book of Mormon open to direct scientific confirmation? Now, there's a couple ways you can address it. The first is no. Now, my wife is also trained in evolutionary biology. She actually has a much better handle on many of the population genetic issues than I do, and I think that she has a lot more insight into this problem. And her point of view is, don't waste your time with this. This doesn't have anything to do with it anyways. The Book of Mormon was meant to be religious history. It was not specifically meant to be confirmed scientifically. Now, this is a point of view you can take, and I would certainly respect that point of view. And I think that there's some very good merits to it. But I don't want to end the discussion there. I want to move on to the next supposition, which is that the Book of Mormon can be tested indirectly. So yeah, that. Does the Book of Mormon make specific genetic predictions that are scientifically verifiable? Okay? This is the crux of the question. And if so, how can these predictions be formed into testable hypotheses? Moreover, how would you design a research program to test these hypotheses? And this is what it really comes down to. Now, again, the point is, there is nobody out there who has specifically designed a research program to get at the authenticity of the Book of Mormon using DNA. It has not happened. It might not ever happen, but certainly it has not happened. This is my argument. The Book of Mormon is in a class of problems that is very difficult to test with DNA evidence. Now, this is because of the nature of the problem. This is not the argument that somehow, because this is a religious history, you can't get at it. You're not allowed to get at it. This is the argument that if you take your word processor and you do a search and replace for the word Lamanite and you put fruit fly in it, you still would not be able to get at this question without great difficulty. And this is because DNA analysis can neither easily refute nor corroborate the lineage history as put forth in the Book of Mormon. Now, there are large classes of questions to which we as scientists would like to have access. There are a lot of interesting things we'd like to get at. Some are easier to get at than others. This question, from a scientific standpoint, is hard to get at, and I will point out why. And I would argue that DNA analysis does nothing to speak to the authenticity of the text. Well, this brings up a critical point now, okay? Let's assume that I'm sitting on the National Science Foundation on a board which is reviewing your research grant. And you have written a research proposal in which you have attempted to test the Book of Mormon and the authenticity using DNA. What are some of the issues you've got to consider? Well, the first consideration is, what is my hypothesis? What scientific hypotheses emerge from the Book of Mormon? Now, I've read the Book of Mormon a number of times, and my response is there are no explicit scientific hypotheses. It was not written as a scientific text. If Joseph Smith turned it into NSF, he would have received no funding. Okay? It is not a scientific document. It was never intended to be. Okay? So what it then becomes is the notion of, okay, if there are no explicit statements of how to test it scientifically, are there any implicit statements? Well, you know, again, to find the explicit statements, one would have to find a verse which reads something like this. And when you shall receive these things, I would exhort you that you would sequence multiple genetic markers from all extant human lineages and verily perform a phylogenetic analysis. And it shall be revealed unto you that Lamanite lineages shall group with the Middle Eastern lineages, and these analyses shall be supported by all forms of data analysis and statistical tests, and he will manifest the truth of it unto you by the power of genetic analysis. And by the power of phylogeny you shall know the truth of all things. Okay? Well, this last part is actually something I teach to my students, but you know, this is an aside. Notice how I removed all the pesky language of praying and spirit and all that sort of stuff. And this is, of course, Moroni 10:3-4, RGE, Revised Genetic Edition. Okay, well, let's get a little more serious here. What are the implicit hypotheses, then, which may come out of the Book of Mormon? Well, I've broke them into two categories, and these are not meant to represent all the possible hypotheses one might pull out, but they're basically two extremes of a continuum where you have to consider when you're testing the Book of Mormon. The first is what I call the global colonization hypothesis. And this is the notion that when the three colonizing groups documented in the Book of Mormon arrived to the New World, there were three of them, and when they arrived, the New World was entirely uninhabited by other humans. Okay, so when they came, they were very lonely. There was nobody else around on the entire North and South American continent. Populations then expanded to fill North and South America, and there was a distinctive genetic signature characterizing the Middle Eastern source population, and that this existed within that population. Furthermore, it supposes that the colonizers accurately carried that pure genetic signature with them. Okay, so you've got a source population in the Middle East, you've got colonizers carrying a genetic signal with them. Colonizers formed a pure and isolated genetic island. Again, that means that when they arrived, there was nobody else around to input DNA. Hence, there was minimal introgression, or what we call gene flow, from any other source population, and that the living descendants of the Lamanite lineage can be identified prior to genetic analysis. Moreover, that the living descendants of the Middle Eastern source populations can be identified prior to analysis. We'll talk more about this in a bit of detail. And finally, that those pesky, complicated molecular events have played a minor or negligible role. Okay, this is one hypothesis you can test, and this is the best case scenario. This is the notion that you've got a few colonizers, they came to America, huge population explosion, no other problems going on with genetics and other input of information from other sources. Can we test this? Well, the hypothesis makes a prediction, and one could argue this prediction, that the descendants of the Lamanites, whoever they might be, and the Middle Eastern source population should carry the same genetic signature. The observation is that modern Native Americans carry a genetic signature suggesting Asian origin rather than Middle Eastern origin. Now, this is a point I'm willing to accept. The modern research on Native Americans thus far seems to suggest an Asian origin. So the conclusion then is that this global colonization hypothesis is incorrect. And I'm willing to accept the notion that this view, this interpretation of Book of Mormon history, does not fit well with current genetic information. I have no problem accepting that at all. But does rejecting the global colonization hypothesis mean that one must logically reject the Book of Mormon? Is this hypothesis the only hypothesis emerging from the Book of Mormon? Moreover, is this the most stringent test of Book of Mormon authenticity? Well, I would contend no, because there's another hypothesis out there, and let me explain this to you. And this is sort of at the other extreme, and this is the local colonization hypothesis. And this again states that there were 3 colonizing groups arriving to the New World, that the New World may have been inhabited by human populations of unknown origin. When these people arrived, there could have been welcoming parties, okay? Population geographic range was much more limited in scope. We're not talking about all of North and South America. We're talking about a more limited geography. A distinctive genetic signature characterizing Middle Eastern origin may not have existed in the source population. Moreover, colonizers may not have accurately carried that pure genetic signature with them. Colonizers may not have formed a pure and isolated genetic island. There was potential extensive introgression, gene mixing, coming in from the native population, potentially corrupting or swamping out the colonizer signature. It is unclear who the extant descendants, the living descendants of the Lamanite lineages might be. It is unclear who are the living descendants of the Middle Eastern source population, and complicated molecular histories and events may have played a major role. Now, I read the Book of Mormon, and it seems to me that this hypothesis is at least as well supported, if not better supported, than the global colonization hypothesis. And a lot of these features, scholars on the Book of Mormon have been saying for 20 years now. Okay, so there's not a lot of new stuff here. Well, here is the problem. Given that hypothesis, what observations would refute it? What would you need to see to demonstrate that that hypothesis is wrong? What sort of data would you collect? What sort of experiment would you design which demonstrates that that hypothesis is wrong? Well, this is the crux of the matter. One could argue that any observed genetic pattern could fit this scenario. And if this is the case, this hypothesis is non-discriminating, and it really is non-testable. Okay, well, let's talk about the complications. I laid this out in front of you now as sort of the hypothesis that we're interested in looking at. Now, again, back to this notion. You are writing a grant. I am reading the grant. If you want to get funded, you've got to satisfy me on some points. And I'm going to lay out 12 points that I need to be satisfied on. Now, this may hurt the heads of some of you. Okay, we are shifting into evolution. Okay, you've heard about us evolutionary biologists before. We are very crafty. You'll notice I didn't say evolution in the abstract, but here you are. You're going to learn a little bit. Which reminds me of the famous bumper sticker which says something to the effect, if evolution is outlawed, then only outlaws will evolve. Okay, so let's not be outlaws, let's evolve together. Number one, was there a unique Middle Eastern genetic markers in the source population? Okay, what do we mean by genetic markers? Well, I tell my students all complicated issues in evolution can be explained with candy. Okay? My undergraduates are very good at understanding things based on candy, so let's talk about some candy. What I've got here are these gumball machines. I've got 4 gumball machines filled with different colored gumballs. The gumball machine represents a population. The gumball is a single genetic marker, and the color is a variant of the genetic marker. Now, when we look at these sorts of data, we're looking at more than one marker, but this is to illustrate the point. Okay. Now, if you have a collection of gumballs which look like this, we refer to this as discrete data. And that's because gumball machine number 1 has only one kind of gumball, number 2 has only one kind of gumball, and so forth. Okay, straightforward, you got it, right? See, it's easy with candy. If we were talking genes, you'd be lost, but it's candy, right? Okay. Well, that's not the way the majority of the data are that we use to infer these historical events within populations. The majority of the data are based on frequency data. And what frequency data means is that you could have 4 different gumball machines, 4 different populations, A, B, C, and D, and they are defined by the frequency of the different genetic markers which are present. So for instance, you might take a look at gumball A and see there's a lot of red gumballs. Machine B's got a lot of yellow gumballs, I think. D's got different colored gumballs. Boy, can you actually see those colors? Okay, beautiful. You're following along, wonderful. Okay, this is called frequency data. Now, the implications of using discrete data and frequency data are very different. And both kinds of data are used in these sorts of analyses, but the preponderance of evidence thus far has been based on frequency data. Okay, so the first point you'd have to satisfy me with is that you had a source population which had distinctive genetic markers. You were able to determine what these are before you go in with your study. Did founder effect skew the frequency distributions of genetic markers? Okay. Sounds fancy, sounds difficult. It's really not so hard. Imagine you were the proprietor of these gumball machines, and you had one gumball machine with a faulty mechanism. So you walked in the morning, and you saw gumballs on the floor. Okay, these are the colonizers. Okay, these are the ones seeking out, trying to find a better life, right? Okay, they're leaving the gumball machine. What is your inference? Where did this come from? It came from B. Boy, you're like my undergraduates. You don't want to yell out and stuff, okay? B. It came from B. And this is fairly obvious, and this is fairly straightforward, right? Well, let's talk about frequency data. We've got our 4 populations, A, B, C, and D. And I've gone through and I've carefully counted the gumballs in each, and we have 4 different colors with different frequencies. Now, can we define these gumballs based on these frequencies? Can we define these gumball machines? Well, certainly you can. You can say that A has got half of them yellow, 25% red, 15% green, 10% blue, and so forth. Okay? So these are the things which are defining our actual populations. Well, let's presume that we have an infinite-sized gumball machine, which is something I dreamed about as a kid. And let's presume that the mechanism is really faulty so that when you wake up in the morning, there's 1,000 gumballs on the floor. And when you count up the 1,000 gumballs, this is what you get. Okay, doesn't it sound like fun research? Yeah, this would be like an ORCA-funded project right here. Okay, so we've got 1,000 gumballs. You count them up. What gumball machine did they come from? Why did they come from C? They closely match that frequency. Why is the frequency not the same? Ask my brother. David. Random sampling. Okay? I love having a statistician in the family, by the way. It's kind of like having a doctor. I don't have to worry about learning statistics because I bring all my problems to him. Yeah, random sampling. Okay? So it's not the same as C. It's pretty close to C. We will infer it came from C. Okay. 100 on the floor. Where did they come from? Okay. They came from A, but the distribution is more skewed. Frequencies are more skewed. Why are they more skewed? Smaller sample. Marvelous. See, you guys can pass evolutionary biology. This is great stuff. Okay? C, 10 of them. There's only 10 on the ground. Where do they come from? Is it starting to blur a little bit? Well, you could say C, maybe. Or maybe you could say they came from something else. Okay, I'm going to give you a hard one right now. 3 gumballs. Where do they come from? You have no clue, do you? Okay, and why do you have no clue? Because you have sub-sampled this population and you have sampling error. And your sampling has been very small. Therefore, the point I'm trying to make here is that undersampling of genes skews frequency distributions, hence obscuring history. That is what founder effect is. Okay? If you have only 2 or 3 people loading up in the station wagon, going to the new world, and they have subsampled this population, how good are you going to be at accurately representing these frequencies? You're going to have a hard time. Okay, let's keep going. Genetic drift. Genetic drift is a similar idea which further obscures frequency distributions. And genetic drift works kind of like this. Assume 1,000 gumballs. Okay, doesn't that sound like a good statistics problem? Assume 1,000 gumballs. Okay, they're here at this frequency. Go in and select 100 gumballs, and you get a different frequency. Fill up a gumball machine using that same frequency. Okay, so you picked 13 yellow ones. Now you're going to fill that new gumball machine with 130 of them. And repeat. What's going to happen to you? What's going to happen to the frequency, guys? It is going to change. You're going to start like this and go to something like that and go to something like that and go to something like that. And in fact, some of these gumballs will disappear altogether because you did not happen to sample them. This is what the original looked like. This is what it looks like after genetic drift. What happened? Frequency distribution was skewed because it further skews the distribution of these genetic markers. Now, you've got to realize that this is a notion which is well entrenched within evolutionary biology. This is how we think new species are made, because we recognize that this has such a devastating effect on the underlying genetic signature of colonizers that they can change sufficiently over a long period of time to make new species. Okay, this is really an important concept. So you got to satisfy me on this. Let me give you the classic example. You have any freshmen here today? Oh yeah, we got one in the back, 1 or 2. Okay. Let me give you an example, see if you can catch on to this. What I want you to do is sit right back and you will hear a tale of a fateful trip that started on this tropic port aboard a tiny ship. Now the mate was a mighty sailing man and the skipper brave and true. 5 passengers set sail that day for, how long was it? A 3-hour cruise. Oh, you guys are so educated. Okay, if this were my evolution class, I'd have you sing the rest of it and then we get up and hug each other and stuff. We won't do that. Gilligan's Island. Okay. Gilligan's Island. Gilligan's Island. My father used to make fun of me for sitting and watching this hour after hour. He didn't realize that this would be the foundation of my science. Okay, Gilligan's Island. Do we have anybody here from Hawaii? Anybody who grew up here in Hawaii? Wonderful. Is that you, Keone? Okay, marvelous. Stand up, Keone. Keone is one of the graduate students who works in our program here. Well, have you watched Gilligan's Island? No, you haven't? Oh, and you think you're educated. Okay, we won't ask you this question. Let me ask you a question. How many people were on the boat? 7. Okay, can you name them all? Well, of course you can. I'll give you some help. Okay, 7 people on the island. Keone, you grew up on Molokai, right? When you went to high school, did you have a name for individuals who looked like this? They're called haoles. Okay. Do haoles form the majority of the population within Hawaii? Not on Molokai. Yeah, we won't talk about Waikiki, right? Okay. Not on Molokai. So my question for you is this. How well do these individuals represent the genetic diversity, if we correlate genetic diversity with, external appearance, how well do they represent the genetic diversity of the Hawaiian Islands? Not well at all. This is skewed. In fact, if you were to allow this population to grow and expand and go for thousands and thousands and thousands of years, so you had the Gilliganites, right? We'd be having this argument about where the Gilliganites came from, okay? Would you infer they came from Hawaii? No, it'd be Southern California, right? Okay. Okay. So this is the point, okay? and sorry to spend so much time on it, but you've really got to recognize that this notion of subsampling populations has devastating effects for how we do this sort of science. Okay, number 4. Did the colonizers arrive to a genetic island? This is the notion that when the colonizers arrived, they could have arrived to where there was no one else around, so that they had the ability to reproduce and carry that same genetic signature. Okay? But what's the implications if they arrived and there were other individuals there? You begin to get introgression, you begin to get genetic swamping. And so what you would have to do is convince me that this is not a problem. How do you account for genetic mixing after the Book of Mormon ends? Okay, we know that the Book of Mormon ends about 400 AD. We've got about 1600 years where there have been other individuals who have input their genes into that population. As a reviewer sitting on an NSF panel, you have got to convince me that this is not a problem. What are the effects of a limited geographic view of the Book of Mormon? Now, the point here is that if you have a limited geographic view, so that we're not talking about North and South America in their entirety, but we're talking about a smaller geographic region, perhaps around Mesoamerica or any place, the notion is you will have a smaller population size. The smaller population size is more susceptible to swamping. You are less likely to find residual populations of these genetically pure Lamanites, whatever they might be. And in terms of genetics, what this means is if you could take a look at the overall picture of the genetics on North and South America, what this might mean is that the colonizers were nothing more than a small genetic blip. And so the issue comes up, can you pick up this genetic blip in your studies? Who are the living genetic descendants of the Lamanite lineage? As a good scientist, as my brother would say, you would have to, a priori, identify who you are going to test in this analysis. Well, who should they be? Well, this is what we've got. We've got an introduction to the Book of Mormon, which says, after thousands of years, all were destroyed except the Lamanites, and they are the principal ancestors of the American Indians. We further have various populations, various groups of people which have been associated as being descendants from Lamanites by various church leaders over periods of time. And we're going to have Dr. Peterson, who can speak to some of these issues in more detail if you're interested in them. Current evidence, as I say, suggests that Native Americans have a genetic history representative of Asia and not the Middle East. But what I want to point out is this. Number 1, the introduction to the Book of Mormon was a commentary. It was a commentary added in 1950. It was a commentary which was based on the best understanding of the brethren at the time. There is nothing within the text of the Book of Mormon which specifically identifies who the genetic descendants might be. Okay, so this is an issue you've got to deal with. You've got to convince me who these descendants might be. Now, this is, of course, a bone of contention. And this is where a lot of the brouhaha has been based on, has been arguing this point. But I don't want to jump into this in any more detail here. Number 8. Okay, we're going up to 12. So I told you it was going to hurt your head, but bear with me here. Okay. Who are the living genetic descendants of the Middle Eastern source population? Everybody is so worried about who are the descendants of these Lamanites. They're not thinking so much about who are the descendants of the source population. Because just as you have introgression and the potential for founder effect and the potential for all these other things going on with your colonizers, you got the same thing going on in your source population. So you've got to convince me that it is reasonable to jump on a plane, go to Jerusalem, sequence a bunch of people, and make the inference that that is what the genetic signature looked like when Lehi left. Okay? And so this is the notion that you are inferring that a modern sampling of Middle Eastern lineages accurately represents the genetic heritage of the source population. How does natural selection influence your results? Now, Dr. McClellan, who is here, is actually a recognized international authority on looking at DNA information and detecting whether selection is present or selection is absent. So pleased to have him here as one of our panelists today. And this is a notion that a particular genetic marker may become more prevalent or less prevalent if it is linked to a feature that natural selection acts upon. Okay? I'm going to give you a more complicated example. Okay, these are jelly bellies. They're more complicated than the gumballs. If you're like me, I go to Costco and I buy one of those great big things of jelly bellies. You ever do that? Okay? They're wonderful things. And you sit there in front of the TV watching Gilligan's Island, and you're eating away blissfully, and then what happens? Oh, you get a cappuccino, you get a jalapeno, you get one of these other very bizarre flavors. Okay. So the notion is, if I had it my way, I'd sit there and pick all these things and throw them out. Okay? And that's essentially what natural selection is doing. Okay, but the problem is, if it turns out that the percentage of jalapeno in your population is what you've used to define your population, and natural selection has thrown it away, then you got a problem, right? Okay, so this is just another problem. How does mutation influence your results? Mutation is simply the notion that we have a certain number of genetic markers we start with, but over time, due to mutations, due to the DNA replicating machinery, you will have changes in those markers. So what this means is that your source population is going to change over time. Your colonizing population is also going to change over time. And this is something we've got to deal with. How does recombination influence your results? Recombination is this notion that you can have two genetic markers in a sexually reproducing population. These can do something called crossing over, and this leaves you with a mixture. And so instead of having just a red and a green, you have two genetic markers which are a mixture of both. Well, the problem with this is that it is not altogether clear when you see that that is what you are seeing. What it can look like is something altogether different. Now, I'm happy to have Dr. Crandall on the panel today because he really is one of the world's authorities on using and detecting recombination within phylogenetic studies. His research has been the most cutting-edge work in this area. And so what does this do? This mixes and confounds evidence of ancestry. Number 12. You guys have been so good. We're on the last one. Hang on. Gene history versus population history. This is the notion that when we use DNA as a genetic marker, what we are really doing is reconstructing the history of that DNA marker. And we are saying that the history of that DNA marker is the same as the history of the organisms which happen to carry that DNA marker. Now, for some very complicated reasons, and we won't get into them here, it turns out that this is not always the case. And we know this. How do we know this? I can do a phylogenetic study on walking sticks, and I can use 5 genes. And each one of those genes can tell me a different story. Some of those genes might tell me more of the similar patterns, some might tell me more different patterns, but the notion is that this kind of naive notion that the molecular data are straightforward and tell you all the same story is really not what the case is. And this is why additional DNA markers and innovation in analysis are required. Modern population genetic studies are moving in this direction where they're taking a look at more information to try and get at these historical things. Well, this brings up what I would like to call the DNA fallacy, and this is number 1. DNA analysis does not require careful experimental design. This is the notion that if I say I have a conclusion based on DNA, I must be correct because I use DNA. And what could be more scientific than DNA? Okay, what you've got to recognize is that the National Science Foundation receives every year literally thousands of proposals from established researchers who say, I will use DNA to do something. And of those, at least a good 50 of them are immediately cut out of the top because a panel will say, no, you really can't use DNA to do that after all. Okay, we recognize this within the scientific community. It needs to be recognized, I think, in the community at large. Second fallacy, DNA provides straightforward, unambiguous, and internally consistent information about the past. As I mentioned earlier, no, it doesn't. Okay? It is not always consistent. It is very often ambiguous, and it is open to further testing. Third fallacy, DNA can be used to infer the genealogy of any organism or any species regardless of circumstance or historical population dynamic. And this is a notion that I can go into a population of fruit flies or whatever I want to look at, sequence every individual and reconstruct their genealogy without consideration of what may have happened in the past, without consideration of confounding factors. Fourth, DNA conclusions are final, decisive, and free of assumptions. That's just not true. Well, let me give you a few facts. Now, the difficulty in giving a talk like this is the last thing I want you to do is walk away with this notion that you can't believe in the science because there's so many assumptions and there's so many things which can go wrong that Dr. Whiting says none of it is believable, you know, let's just all go home. You know, might as well call this the psychic network is use DNA, right? But I'm emphasizing the limitations simply because the limitations have been left out entirely in this discussion of whether DNA tells us anything about the Book of Mormon. Well, what are some of the DNA facts? It is an incredible tool for inferring history. My research program is based on DNA precisely to infer history. But experiments need to be properly designed, hypotheses need to be formulated, assumptions must be tested, analyses must be appropriate for the data at hand, and the conclusions are the best current estimate, but open to revision with additional data and or analytical tools. Let me drive the point home for you. Okay, fruit flies. Everybody loves fruit flies, right? We did this in beginning biology. Let's assume we were testing the hypothesis that 4,000 years ago there was a wonderful fruit fly here in Provo. You can tell it's here at Provo because it's holding the BYU flag. One day, a large windstorm comes and blows it, so it lands on the home of Keone in the Hawaiian Islands. This is a very blissful fruit fly now because, if you had your choice between living in Hawaii and living in Provo, there's no competition. Okay. As a good fruit fly does, it reproduces and makes more fruit flies by itself. Okay. However, there are also the native fruit flies in their native fruit fly grass skirts, which are there to meet these fruit flies. And these things are reproducing with each other, okay? And we're looking at this over a couple thousand-year period. But not only that, you have continual fruit flies being blown in from all over the world, okay? So they are adding the genetic information to this population. My question is, could you find genetic evidence of the Hawaiian colonization event? My answer is maybe, okay? You could get lucky. The population dynamics might turn out that you find it. That particular genetic signature might have survived. You might have some new method of analysis which allows you to tangles all of these problems. But the fact that you did not find that genetic signature would come as no surprise to biologists who do this sort of research. Why? These are tough questions. And the oversimplification of these questions has done nothing to help this discussion and this debate. Okay, so what critics of the Book of Mormon would have you believe is that testing the DNA via testing the Book of Mormon via DNA is as simple as lining up gumballs to the machines they came from. My argument is this: if you were to take a look at the underlying pattern of genetic history, you would have a Jackson Pollock. Okay, this is called Jackson Pollock. This is, I believe, his work called Autumn. Now, is there a pattern in this? Well, yeah. Okay, if you have a very tuned eye where you can see this as an art critic, you might look and say, there is a marvelous pattern there. Is that pattern altogether obvious? Not to my eyes. Are there certain assumptions you need to look at to get at that pattern? Well, you certainly do. And this is the crux of the problem. We are trying to figure out a problem which looks something like this, and we're using means which are not well adapted to get at these kinds of problems. So again, what I'm not saying is that all population genetic studies are bogus. Okay, if you go home and you think that Dr. Whiting gave a speech saying, don't trust DNA, don't trust population genetics, don't trust phylogenetics, you're wrong. I'm not saying that at all. I am not saying DNA is an unreliable tool. I am saying it is a tool. Like every other tool that a scientist uses, it has to be wielded properly. I don't want you thinking that the science has so many assumptions that the results are not believable, but what I am saying is this. The local colonization hypothesis is hard to test because of complications associated with the Lamanite lineage history. And in particular, to me, the things which are most striking are the problems of founder effect, drift, and the potential for extensive introgression. And again, as I say, it's not an argument because, they're Lamanites and Nephites. It's what the DNA is saying. Okay, DNA doesn't care whether it's in a fruit fly or in a Lamanite. Okay, it acts the same. DNA evidence is not likely to unambiguously refute nor corroborate this hypothesis. And this is an important point. This hypothesis has not been specifically tested. There has been no study which the critics have put together. They have not gone out and obtained research grants. They have not designed studies. They have not accumulated the data. They have not specifically tested this question. This hypothesis has not been tested. DNA evidence does nothing to speak to the authenticity of the text. And I want you to know, and I say this in all honesty, I would be just as critical as anyone who rose up and said, I now have DNA evidence proving the Book of Mormon is true, as I am of the critics who say we have evidence that it proves it is not true. Okay, the science is tough, and these answers do not come unambiguously. You can't claim it's scientific if you ignore the science, and that's the take-home point. Okay? And I thank you for your attention. I want to leave you with this quote from James E. Talmage, and I think we'll take a break for a moment. Thanks.